I did my project on Monica Bettencourt-Dias. Monica Bettencourt-Dias grew up in Portugal and was born in 1974. She got her undergrad degree in biochemistry at the University of Lisbon. She entered her PhD program at the Instituto Gulbenkian de Ciencia and then completed her studies at the University of London. She then enrolled at the University of Cambridge and Birkbeck College, where she focused on postdoctoral research in kinesis. She is very involved in the science field outside of research, where she has served in multiple leadership positions, and she's also a public supporter of other scientific groups, including the IGC and EMBL, and outside of science, she enjoys listening to music, photography, and traveling. I identify with Diaz because she works in two fields I'm very interested in, and her topics of research are also very interesting to me. She's also a very passionate person and dedicated to science and people, where she interacts with the social community and is very nice to people overall. Bettencourt-Dias's main focus of research is dedicated to centrioles and their role in the body. In the past, she has targeted how they are involved in multiple processes, including cell division, disease development, and cell cycle regulation. In her most recent experiment in the past 20 years, her most notable experiment, Diaz performed an experiment to determine the role of the SAK-PLK4 kinase in central development and how that affects different functions in the body. Kinases are a part of the polo family, responsible for regulating cell division and assist in mitosis and cytokinesis. SAK's role includes assisting in mitosis, gene regulation, and development of organelles. In Diaz's first part of her experiment, she focused on how the reduction of SAK affects central structure and function. She depleted 70% of SAK mRNA in Drosophila and allowed cells to divide, and results showed that structures and division weren't organized. Some results included the loss of Y-tubulin, which are centrosome markers in cell division, and frayed meiotic spindles. And as you can see in the diagram, the GFP is our control, and you can see that cell division is very controlled in all three panels, but in both the mutated SAK and the mutated polo, you can see that the fray is either widening or completely frayed, and you can see that in all panels across SAK and polo. In Diaz's second part of her experiment, she wanted to know if SAK-PLK4 was required for centriole duplication. She exposed Drosophila cells to dsRNA and allowed them to divide, and after 16 days, over 93% of cells had no centrosomes. And in order to determine why, she examined spermatogenesis in affected PLK males. She observed that spindle poles were either very broad or had absolutely no astromicrotubules, and this caused sterility in the males that were affected as well. These observations were associated with defective chromosome segregation and cytokinesis. In the third part of her experiment, she also wanted to know how it affected human cells, so she again reduced SAK transcript by 70%. She found that HeLa cells, which are cells grown in a lab with one centriole, increased tenfold, and cell apoptosis over doubled. She also observed that spindle abnormalities and shape changed drastically in affected PLK cells. And as you can see in the diagram, many abnormal spindles, multipolar spindles, monopolar spindles, and other affected spindles are in this picture. And you can see in the chart below that the amount of abnormalities in spindles increases tenfold. And this concludes that SAK kinase is required for central duplication and reduplication in HeLa cells. What's been uncovered in the past, Diaz has been interested in, quote, the accounting, assembling, and maintenance of centrioles, cilia, and centrosomes, and their variations observed during development, evolution, and disease. She also has discovered many different things, including mechanisms that regulate centriole biogenesis through feedback loops, and other discoveries mentioned below. She hopes to uncover more about how centrioles affect cell regulation and development in her future research. As a whole, Diaz is a very accomplished scientist where she focuses a lot on centrioles and cilia, but she's also very passionate and dedicated to what she does and stays very involved in the scientific community today.